Anil Dewan, PhD, is the chairman and president of Nanoviricides. He is also the inventor of Athera core technology that enables nanoviricides drugs. Dr. Dewan has extensive product development experience while raising financing from collaborations, SBIR grants, and other revenues. Dr. Dewan?
Thank you very much. I did a presentation in the morning today, and Brett had said that the audience in the afternoon is somewhat different from the audience in the morning, and he didn't want people to miss out on this presentation. So I'm very grateful to him for this second opportunity. I do see a lot of new faces. We are a public company already, so I have to give you a disclosure statement as to that we are a public company and that there are risks involved. I'm going to give you a corporate overview in what I call the micelle. Basically, normally you would say nutshell. What we are developing are micelles, so I use the word micelle. It's also a globular container, so it works well. We are a public company. We have 6 different antiviral drugs in our pipeline right now, which address about $40 billion market space according to 2013 projections from a 2009 market study. That's probably a 60 billion market space if you look at it today. We have a novel mechanism of action, which is always a sought out goal in virology especially. And because it's a novel mechanism of action, all of our drugs are first in class drugs against every target that we choose. These are typically the things that pharmaceutical industry people want to hear. Is it novel? Is it first in class? Okay, we are. Yeah. And what we are developing are intelligent nanomachines that use the virus's own smarts to defeat it. And if you looked at the logo before, it shows circles. The inner circle represents the virus, and the outer circle represents our material. Basically, our material goes at it and slams on the virus. And that way it defeats the virus. We have a strong patent protection. It's a platform technology, and it enables us to develop new drugs against different viruses very rapidly. First 6 years, we developed 1 new drug candidate against a new viral disease, 1 per year. After that, we focused on developing further on influenza as our financial situation improved with the raises that we have done. And now we are going further with influenza into making it IND ready. There are 2 different influenza drugs, and I'll talk about it later. The one thing about this technology that you want to learn is the phrase resistance is futile. From the virus's perspective, today, the humans, our resistance is basically futile when you talk about influenza, when you talk about HIV, when you talk about the MERS virus that is going on, any novel virus. And the viruses know how to defeat it because A, they are intelligent nanomachines themselves. They have built mechanisms for defeating our immune system and hijacking it and do whatever they want. B, they mutate constantly. They change all the time. Okay. And because they change, the drugs that we develop, the vaccines that we develop, they don't work. They will work today when we have developed them. 3 years later, classic case with influenza, the resistant strains develop, the resistant strains then start taking over. And what we are showing is we are developing, so this was the major problem that I started working on, is how do you correct against that? And the counter against that is to use the virus's own smarts. No matter how much the virus changes, it still lands on exactly the same receptors on the cell, and it uses exactly the same kind of contact mechanisms with the cell surface, with those receptors. So that information does not change. And so we use that information, and what we do is we make small chemicals. We design these chemicals based on that information. We display them on the surface of the polymeric micelle, and then that looks like a surface of the cell to the virus. And it lands on it, and then it thinks it has landed on its target. It starts dismantling itself, because it is programmed to dismantle itself after it goes inside the cell. So that's the technology. There are, of course, once you go deeper into the science, there are a lot of nuances, lot of details that we have, of course, designed and developed. I'm not going to go into that, but this is the overview of the technology. And because of doing this, each drug that we develop is broad spectrum against that particular virus. When we talk about influenza, we don't say, okay, it will work against this one and it will not work against that one. And we have to prove that, obviously. And we have proven that, and I'll get to the data, hopefully. The other thing about this company, it is a pioneer in polymeric micelles in nanomedicines, and that I already told you. Not only that, it is the first company in the world to have developed a nanomedicine that can be delivered orally. We can make a cough syrup, we can make it into a tablet or something like that. That has not been achieved yet. There is a lot of work going on nanomedicines today that has never been achieved yet, and we made it happen. So, and we have a strong team now with an independent board of directors. As you all know, independent board of directors is important. We are currently trading on OTC bulletin board, and it is an important requirement for us to be uplisting to our next exchange. So, the current state of development, I alluded to some of this before. We have two different drugs that are in IND enabling studies. Injectable flucide, which is for hospitalized patients. It's a high activity, but it is injectable. Okay. Then oral flucide, which will be developed probably a year beyond that. After that will come out, and that is also in IND enabling studies. More studies are needed for oral drug in this case because it's novel. And for both of those drugs, we are using the same information that we learned from FDA, that we learned in the pre-IND meeting we held in March 2012. So we are going ahead, we are moving slowly, but definitely towards the goal. The other important thing that we did not have rather, is manufacturing capability. These are novel materials. For any novel material, manufacturing capability is important, and it becomes a risk factor. For us, because we were driving in with a new kind of polymeric chemistry, it was an additional thing. So what we decided to do eventually, after trying to do it through contract manufacturing for a long time, for about 4 or 5 years, we decided to bring that in-house. And that is taken up as a separate project under a private company, and Nano will be able to lease that facility. That facility is in development. It will be available to us in end of this year, December 2013, and then everything ramps up. Because once that facility is in hand, we have to make the materials for IND studies. Once we have made one drug going into IND studies, for us, it is just basically replicating the same formula for the other 6 drugs, remaining 5 drugs. So process scale up, CMC work, that is also in progress. Some of you may know what that means. Financially, we are doing very well right now. We are at the best state that we have been in our history of 7 years now. We have raised only about 42 million since 2005 in the 7 years. And out of that, we are still holding 16 million in hand. We have more than 2 years worth of amount of cash that we burn in hands. We only burn about 4 to 5 million a year in terms of cash. And in that, only about 26 million spent, we are sitting with 6 drug candidates against 5 different diseases. And I have an expert from pharmaceutical industry here. He will tell you what a phenomenal achievement that is. So we have enough money to be able to take flucide, one of the 2 flucide drugs, all the way through phase 3 if we have to. We don't believe that we will have to. We believe that we are making progress in the other side also in terms of dealing with pharmaceutical companies and being able to license the drug. But if we have to, that is not an expensive drug, and we have enough cash in hand for doing even that. Okay. So, and that is based on our discussions with CROs as to what is needed, and our pre-IND meeting as to what is needed for the clinical trials. We have a registered shelf that is effective, that has 55 million still available in it. So raising money, if we need to, is not going to be an issue for us either. So basically, we believe we are now no longer what it was 2 years ago, that it was a teetering company that is developing something completely novel but doesn't have the money to do it. We are no longer there. We are a stable company, sort of like what a VC funded company usually is on day one. We have achieved that state in financial terms, and we are going further from that. We have about 163 million common shares outstanding. The stock price was about 64-65 cents today. We have an independent board of directors with very marquee people. Professor Milton Buniat is the ophthalmologist, surgeon, chair professor at Baylor College of Medicine, Caroline F. Ellis Chair. And obviously, when we go towards clinical trials for our eye drug, which I will show you, we hope that he will be one of the critical points that leads it. He has been a major investor in our company and a cheerleader as well. I mean, when even a person like me who does science in the lab and then finds that there are obstacles on the outside world to bring that science out, he has been a cheerleader, his sister has been a cheerleader, both of them have been major investors. So that kind of a marquee person we have on the board. Professor Mukund Kulkarni is Chancellor of and Professor of Finance at Penn State University. Strong integrity, strong administration capabilities, a strong growth-oriented view. He has a huge growth plan for the university that has been implemented. And so those are the kind of things that we need when we experience the J stick going into clinical trials and getting the approvals. So we are putting that together. Stan Glick is a CPA and a well-known member in the community, public auditor, and he's the chair of the audit committee, and the executives you already know. So the 6 drugs I talked about, 1 injectable for influenza, 1 oral for influenza, eye drug, which is going to encompass basically 100%, 99% of the viral diseases of the eye. We are going towards that. HSV, herpes simplex virus, cold sores, genital sores. HIV, the drug that we developed against HIV phenomenally works almost 30 days and controls the viral load for 30 days after the drug has been withdrawn, after we stop treatment with that drug in animal studies. These animal studies, by the way, what we are doing are directed against the virus. So these animal studies that we are doing, the animal is just a test tube. The animal system is not involved. So they are translatable straight out into human studies. And of course, I talked about MERS coronavirus briefly. I can talk to you about that. Yeah, we can. We are already working on that. Like as you will see, there is one small chemical that we have to develop and design, and if that works, we have a drug against it. It's a very, very quick development cycle that we can bring to the market, once we are there. So I already talked about the market size. The technology, our nanomedicine approach leads to well-defined chemical entities. It's a polymer material, but it's a well-defined chemical entity. The ligands are regularly spaced at regular intervals along the polymer chain. And again, the pharmaceutical people will know how critically important that is. They have extremely high efficacies and very strong safety profiles. One of the reasons people ask me why we haven't started the tox package studies, the problem was we need very large quantities of drug. So we might as well bite the bullet, do the process scale up first, and then go for it. That's what we are doing. How do we do this? So this is a cartoon of HIV, and what you see here, there are two different. There are two different receptors that HIV uses, CD4 and CCR5 are the usually ones. CCR5 can be substituted by CXCR4. CD4 interaction is the first one and the most important one. That interaction, no matter how much HIV varies and mutates, that particular interaction doesn't change. So we mimic that. We developed small chemicals. Initially, we went with peptides, copying exactly the same loop, and we improved upon that. And then what we do is we take these small chemicals, we attach them, and you see that cartoon. Now that surface, those triangles and rectangles are flags to which the HIV binds. So that surface looks exactly like the cell surface to the virus, and it binds to it. So now, even if the HIV virus mutated, it still has affinity to our nanoviricide. It's going to bind to it. So our drug is going to be effective. That's the whole hypothesis. Attack the virus, that gives you inherent safety. Okay. Bind, encapsulate, destroy mechanism. This is going beyond immunotherapeutics. Antibodies and vaccines are well-established therapies. Antibodies bind with 2 points. Nearly about 10 antibodies have to bind to a virus particle before it is neutralized. With one micelle that we have, once it comes into contact, it will be neutralized because there is that much binding capacity in that, like the cartoon that I showed you. And it's a biomimetic. Looking at the other way, how does this nanomicelle work? And you are seeing the steps 1, 2, 3, 4, how in the step 1, single interaction has taken place, step 2, multiple interactions have taken place, and then the interior of the micelle, which is lipidic, is now fusing with the exterior of the virus coat, which is also lipidic. It's a lipid membrane. So once that fusion takes place, the virus is completely coated. It's the proteins that it displays, the so-called glycoproteins, it displays on its surface for binding to the cell surface receptors, are now either covered, bound to the ligands, or simply uprooted from their location and not available. So the virus is not infective either. We are a field leader in intellectual property. I'm going to skip this slide. We have over 18 patents that we have issued now. We have got broad claims coverage as written in the United States. And that's another feat that is very difficult in today's environment. We recently raised $6 million from 3 family offices and charitable foundation. I already talked about the 16 million we have in hand. Okay. Now, here is where we can show you some demonstration of the principle. We used just the bare polymeric micelle and attacked MCMV, which is a multi-capsid virus, with it, and we captured the intermediate state, which is on the right, the A label. You can see the capsids are slipping out slowly, and the bright area, that's where all the lipid coat is congealing together. Possibly that's where the nanomicelle is pulling it there. Okay, so when we use the once with the virus binding ligand, CMC also binds to the CMCV also bind MCMV also binds to the same receptor, sialic acid receptor, so that influenza binds to. So we were basically using that one. When we did that, we always were getting this C type of structures where the capsid is floating and you cannot find any evidence of a entire virus particle. Okay. So it does, at least in some cases, this material does destroy the virus particle. Against influenza, I will stress that we have already shown, and I will show you the slides now, H1N1 and H3N2. It's always been heavily superior to oseltamivir in all parameters. We are using survival as the objective endpoint, so we are using highly lethal models, unlike other people's studies. Okay. We show lung viral reduction as well as lung protection. Here you see PO means oral, and you are seeing against H1N1 and against H3N2. You are seeing how significantly high the survival level with our drugs was against Tamiflu, and Tamiflu was barely superior to no drug. Yeah. So that is a clear demonstration. This is the same thing in viral load. And the injectable HIV study, injectable influenza study, people have seen this before, and we had achieved complete 21-day survival of the virus, survival of the mice in that study. And the market, of course, is huge, especially when we open up with the oral. We believe that then the entire 6 billion population will be open to take our drug, and imagine the market size for that. With this, I'll stop my presentation.